Okay. So hello everyone. My name is Anirudh Belwalkar, and today I will be presenting my work titled Evaluation of SIF Retrievals from Narrowband and Sub-nanometer Airborne Hyperspectral Images flown in tandem, modeling and validation in the context of plant phenotyping. This work was conducted as part of my ongoing PhD project and has recently been published in the Remote Sensing of Environment. So the content of today's presentation will be based on this published paper. I would also like to thank my supervisors, Pablo and Thomas, along with all co-authors, Andrew, Alberto, and Rocio, for their contributions in this work. About 78% of the energy incident on the plant leaves is absorbed, and the remaining energy is either transmitted or reflected. The absorbed energy cannot be fully utilized for photosynthesis, and the excess energy is partially dissipated in the form of heat and partially re-emitted in the form of an electromagnetic signal that gets superimposed on the reflected radiance. This signal is called solar-induced chlorophyll fluorescence, SIF, and it constitutes a very small fraction of the total incoming solar radiance. Due to its close link with photosynthesis, SIF serve as a direct indicator of pre-visual vegetation stress and can provide useful information about plant photosynthetic activity. SIF spectra spans in the 650 to 500 nanometer region with two maxima, one around 685 nanometer and other around 740 nanometer. Specialized algorithms are required to decouple its contribution to the reflected radiance because of its very low contribution to the total reflected radiance. These algorithms mainly use discrete solar or telluric absorption lines of the spectrum because at these particular features, the relative contribution of SIF to total radiance is higher. The oxygen absorption bands O2A and O2B, centered around 760 and 687 nanometer, are most commonly used for SIF quantification because these are wider and deeper than other absorption features. Spectral resolution, signal to noise ratio, and spectral sampling interval are the key parameters that influence the accurate estimation of SIF, because these parameters directly influence the shape and depth of the absorption features, which are required for accurate SIF estimation. In the current slide, the effect of spectral resolution on the radiance signal in the O2A absorption feature can be observed for different combinations of spectral resolutions using floor cell 3 simulations. As the spectral resolution reduces from 0.1 to 5 nanometer, a decrease in O2A absorption feature depth can be seen, and along with that, an increase in the absorption minima can be observed. These simulations, by these simulations, it is clear that sub-nanometer resolution, which is with full width at half maximum of less than 1 nanometer, enables the proper identification of absorption features, which is required by the SIF retrieval algorithms for accurately quantifying SIF in absolute terms. Although sub-nanometer resolution is recommended for accurate SIF quantification, several studies using narrowband imaging and non-imaging sensors with 4 to 6 nanometer FWHM have shown that the relative SIF variability captured by these narrowband sensors can be a valuable plant trait in variety of contexts, including plant phenotyping, plant physiology monitoring for detection of biotic and abiotic stress. Nevertheless, proper interpretation and validation of such narrowband SIF estimates are required through its comparison against sub-nanometer airborne SIF observations. This assessment is critical, especially for studies that use SIF in relative terms as a proxy for assessing the plant condition. As a response to this, the objectives of this research were to assess the reliability of airborne narrowband hyperspectral imager with 5.8 nanometer FWHM in capturing the within SIF variability of the experimental field through its validation against a sub-nanometer resolution hyperspectral imager with 0.1 to 0.2 nanometer flown in tandem across plant phenotyping studies. The second objective was to assess the quantification of these absolute SIF levels quantified through the 5.8 nanometer FWHM hyperspectral imager and its comparison with the simultaneously acquired absolute levels of ground-based and airborne-based sub-nanometer SIF estimates. So the two hyperspectral imagers used in this study were onboard a Cessna aircraft operated by the Hypersense Lab, University of Melbourne's Airborne Remote Sensing Facility. The spectral characteristics of the two hyperspectral images can be observed from the slide. The effect of spectral characteristics of the two hyperspectral images can be observed in the sample radiance spectra collected from the same vegetation target. The transparent gray box in the narrowband imager radiance shows the region covered by the sub-nanometer imager. The study was conducted at two different wheat and maize phenotyping trials grown under different nitrogen application rates over 3 growing seasons in Victoria, Australia. The details of the experimental sites and the airborne campaign are provided in the given table. So an additional experiment was conducted at experiment number 3 for evaluating the impact of sensor altitude on the SIF estimation. So at 3 different altitudes, SIF was estimated. And these acquisitions were completed within a 20-minute time interval to minimize the impact of sun sensor geometry and the changes in absorption, changes in the atmospheric conditions affecting SIF. Here the figure shows the imagery collected from the rain-fed wheat experimental field at the study site 1 and the irrigated maize experimental field at the study site 2 and the locations of the corresponding nitrogen treatment experiments. Now the methodology for airborne SIF quantification. From the narrowband and sub-nanometer airborne radiance imagery, mean radiance was calculated for each plot by computing the average pixels within the whole region of interest, excluding the boundaries and non-vegetation pixels. The incoming irradiance at the time of flight was calculated using Ocean Optics HR2000 spectrometer with 0.065 nanometer FWHM. To match the spectral characteristics of the individual narrowband and sub-nanometer hyperspectral imager, the irradiance was resampled based on the spectral characteristics of both images. Using this resampled irradiance and radiance from the two hyperspectral imagers, SIF was quantified using 3FLD method. SIF was further corrected based on non-fluorescence targets to account for atmospheric and calibration effects. Finally, the corrected SIF was normalized to a nadir viewing angle using a recently proposed angular normalization approach, which is based on NIRV, to account for the directional effect of SIF. At experiment 3, leaf canopy level measurements were also made at each plot for calculating ground-based SIF, which served as an validation for the airborne SIF. Apart from these spectral measurements, leaf level measurements were also conducted at all the 3 nitrogen treatment experiments for measurement of chlorophyll, nitrogen, balance index, anthocyanins, flavonols, and steady state chlorophyll fluorescence using the leaf clip sensor Dual-X and fluoropen simultaneously with the airborne campaigns. Apart from these experiments, a simulation study was also conducted using scope model to evaluate the influence of spectral resolution on 3FLD based SIF quantification. A total of 400,000 scope simulations were generated, and the input ranges for these input parameter ranges for these simulations were used using the existing literature and the leaf level measurements taken at field site 1. The default 1 nanometer spectra from the scope simulation was then resampled to 5.8 nanometer FWHM to match the spectral characteristics of the narrowband hyperspectral imager. Then, for each airborne radiance spectra at experiment 1, we selected 10 closest simulated spectra using root mean square error as a cost function. So the figure shows on the left shows the airborne imager collected spectra in the field, and the right shows the finally selected simulated spectra based on root mean square error cost function, which were finally used for further analysis. Apart from this, additional simulations corresponding to spectral resolutions of 2, 3, 4, and 5 nanometer were also generated, keeping the default 1 nanometer SSI of scope simulations. Now the results. So in experiment 1, leaf physiological traits were significantly different across plots fertilized with different application rates. Whereas for experiments 2 and 3, differences were non-significant, but there is a visible variation in the leaf physiological traits. For fertilization rate was positively associated with the chlorophyll and NBI, whereas a negative trend was observed with flavonols and anthocyanins. These leaf physiological trait results suggest that the nitrogen treatment applied was sufficient for providing variability across the experiments. So the shape and depth of O2A absorption feature and O2B absorption feature were affected by the spectral characteristics of the two hyperspectral images. Due to the coarser resolution of narrowband imager, the O2B absorption region or the O2B minima cannot be identified. Whereas for the O2A absorption region, a decrease in the depth of O2A absorption feature and an increase in the radiance minima can be observed. These results are in agreement with the fluorescence 3 simulations shown earlier, which showed the effect of spectral resolution on the radiance spectra. Now for experiment for study site 1, despite differences in the absolute depth captured from the two hyperspectral images, a significant relationship was obtained between them with an R square of 0.9 when using a full data set with more than 100 plots across the whole experimental field. Although SIF narrowband and SIF sub-nanometer showed a lack of correspondence with RMSE of 3.86, they were highly correlated. They were significantly correlated across with an R square of 0.85. Now the results corresponding to the 3 nitrogen treatment experiments. So the absolute SIF levels quantified by the SIF, both hyperspectral images were affected by the spectral characteristics, as they showed the root mean square error in the range of 3.28 to 4.69. However, for all the three experiments with different nitrogen treatments, a significant relationship between SIF captured from the two hyperspectral images were observed. These results demonstrate that the SIF obtained from narrowband imagers is quite reliable when compared to the SIF sub-nanometer. However, the absolute value is still overestimated. Now the SIF comparison across different altitudes. So for narrowband imager, 2200 meter altitude was excluded because the size of the pixel was too coarse relative to the plot size. Both the relative depth and SIF showed significant differences with increasing altitude. With the depth of O2A absorption feature increased while the SIF decreased with sensor altitude. At both 900 meter and 1200 meter altitude, SIF were significantly correlated across the two hyperspectral images. However, SIF at 900 meter showed a higher root mean square error, which can be attributed to the higher SIF levels obtained at lower altitudes, which were shown in the earlier slide. Similar results were obtained when SIF at different altitudes was compared from the same hyperspectral imager. The top figure shows the SIF at 3 different altitudes from the sub-nanometer, and the below one shows for the narrowband imager. Strong correlations were obtained at all the different altitudes, across all different altitudes. However, as shown in the previous slide, with altitude, the SIF values tended to decrease. So previous studies have shown that the depth of O2A absorption feature increases with sensor altitude. So this is consistent with our study, which shows that with increase in depth, with increase in altitude, depth increases for both hyperspectral images. Despite correcting for atmospheric effects, SIF decreased with increasing altitude. This decrease can be linked to the applied atmospheric correction method, which relies on non-fluorescence targets like bare soil. Since the relative increase in O2A absorption depth for vegetation target is higher than the relative increase in absorption depth for bare soil targets, there is a bias in the corrected SIF values, which is the reason for different SIF values at different altitudes. Now the validation of both airborne hyperspectral imager quantified SIF with ground-based sensors. So for at all 3 altitudes, for sub-nanometer imager, the SIF quantified was well correlated with the ground-based SIF with R square higher than 0.9 in all cases, 0.9 or higher in all cases. Especially for 900 and 1200 meter, a good correspondence with ground-based SIF values was obtained with root mean square error less than 0.1. However, for higher altitude 2200 meter, there was a lack of correspondence due to the altitude which I explained earlier, due to the bias effect. Similar results were obtained for the narrowband quantified SIF validation, where at both 900 and 1200 meter altitudes, the root mean square, the R square was higher than 0.85, showing a good strong relationship. Now the results from the scope modeling experiment. So low resolution SIF from 2 to 5.8 nanometer were well correlated with the reference SIF at 1 nanometer. With coarser resolution, the RMSE value increased. As you could see, the RMSE was highest for 5.8 nanometer and the lowest for 2 nanometer SIF. The offset of the linear relationship between the 3FLD relationship increased with FWHM from 2 to 5.8 nanometer, while the slope remained constant. These differences in SIF values based on the spectral resolution of the sensor are in agreement with the results that were obtained for the experimental study. And finally, we compared SIF, narrowband SIF against vegetation indices, EVI and MTVI2, which serve as a proxy for canopy structure to evaluate if the narrowband SIF is driven by the changes in canopy structure caused due to nutrient variability. both vegetation indices show weak and non-significant relationship with narrowband SIF, which implies that narrowband SIF is not was not affected by the canopy structure, and the variability observed in SIF was due to subtle changes in physiology across the different treatments. As conclusions of this study, SIF comparison results from two hyperspectral images with different spectral resolution flown in tandem shows a statistically significant relationships with an R-square value of higher than 0.75 for 3 different growing seasons and for 2 type of crops. So in precision agriculture, plant phenotyping and plant physiology monitoring experiments, the focus is on assessment of relative SIF variability across the experimental field. So for such type of applications, narrowband SIF might be of interest. The SIF variability was not affected by the changes in the crop structure, but was due to the subtle changes in the plant physiology. And additionally, the lightweight nature of these narrowband images makes them suitable to be used on board the drone platforms for precision agriculture applications that require high spatial resolution data for mapping the SIF variability across the experimental field. So since the SIF estimates from these narrowband hyperspectral images are overestimated, their interpretation becomes very critical, especially for plant physiology monitoring assessment. Therefore, our current work focuses on taking the spectral resolution of these narrowband hyperspectral images in account by using approaches based on radiative transfer modeling and machine learning to scale the narrowband SIF to finer spectral resolution. And finally, a preliminary result from the scaling study, where here you could see the validation of the proposed approach for scaling narrowband SIF estimates to 1 nanometer FWHM using scope and machine learning methods at plot scales. So the reference SIF was obtained using the simultaneously acquired sub-nanometer resolution spectra, which was resampled to 1 nanometer FWHM for comparison. So the estimated SIF was able to map the spatial patterns of SIF across the experimental field when compared to the reference. As you could see here, the low SIF levels were captured by the reference can also be seen in the estimated ones. Similarly, the high SIF levels here, which showing it dark yellow, can also be observed in the estimated SIF at 1 nanometer, and this is from the narrowband imager. So these results seems promising, and this methodology will be further tested for the second site to test its robustness. Thanks everyone for paying attention.